Hello everyone. Welcome to the second talk in today in the colloquium series. Today we have Robert Young from Rutgers University. He's going to talk about the twists and turns of circular DNA. Um, for those of you who will have questions, and that is a good thing, please raise your Zoom hand at the end of the talk before asking your question. So I'll hand it over to you, Rob. You can go ahead.
Thank you very much. Good afternoon, everybody. As my host mentioned, my name is Robert Young. I am a graduate student actually at Rutgers University. I'm working on a second master's in physics right now, as well as my PhD in chemistry. And I'm excited to talk to you today about some of my dissertation work that I'm doing under Dr. Wilma Olson in our biopolymer structures lab. The talk is entitled Twists and Turns of Circular DNA, the biophysical implications of protein DNA interactions. I am an alum of Appalachian State. I graduated in 2011. And when I was navigating Appalachian, I ran into a little bit of a problem where my interests didn't really align with any of my cohort. At the time, there was a lot of engineering students. There was a lot of engineering and professional science master's students, as well as astronomers. And I didn't want to go in that direction. I was really interested in what other directions you could go with in physics. And I really enjoyed chemistry. I really enjoyed biology, and I just have a love for mathematics. So I worked with Dr. Chris Saxon on what other directions that we could go in terms of my study. And I always kind of came into the same direction in terms of what's occurring in the cellular environment. Because to me, proteins and DNA and RNA, they all interact really well, but we tend to forget that they don't have brains. Why they are doing what they do is simply based on electrochemical gradients that exist in the cellular environment. But there's still a lot of work that we can do to help understand why certain things are occurring in the cellular environment. And we can use physics to help understand and work with exploring these interactions. One of the ways that we can look at this is just the way in which DNA is traditionally viewed and organized. It starts out as the rigid form that we were all taught in elementary school, where you have DNA as a double polymer that wraps around in this helical form, and it has a lot of these base pairs like adenines and thymines and guanines and cytosines, and they all base pair to each other in this ladder configuration. And it starts to increase in levels of organization, such as the second level that you see here, the beads on a string. Then the third level is going to be, this 30 nanometer chromatin fiber. And then it keeps going to a higher level of organization. I love this picture only because it reminds me of just how ridiculous it gets when you don't pay attention to your headphones. It's the same type of organization. It's this coiling that's going to occur. And it's this higher order packing, so that once it becomes this coiled structure, you have better packing in the smaller volume. But chromosomes are not the only type of form that exists for DNA, especially in higher ordered organisms like humans and mammals and whatnot. There are also circular forms of DNA that are common in our biology, and it's not really something that's talked about a lot unless you go into studies like molecular biology courses, virology, and parasitology. Viral DNA is typically organized as closed form circular forms of DNA. When we talk about circular DNA, what we're really saying is that the two ends of our DNA are covalently closed together. Sometimes it takes form in needles where it's highly coiled on top of itself, and sometimes it is relaxed. But when we talk about circular, what we're really saying is that the ends of our DNA are combined together. Additionally, mitochondrial DNA is organized as interlocking circles of various sizes. You might have large maxi circles that will have a lot of smaller mini circles that are attached to it. One type of study that was being done on a parasite that has a mitochondrial piece of DNA that is notoriously associated with African sleepy sickness. And we want to look at understanding why different forms of that, different sequences of forms of the circular pieces of DNA contribute in different configurations. But also, circular DNA is found in targeted genomic expression in cancer cells. If biology has taught me one thing, it is that it wants to spend the least amount of energy as possible. And it takes a lot of energy to get protein into the nucleus, find the gene it wants on the chromosomes, adhere, express, and go forward. If it's going to be a commonly used gene, you could take that gene, put it into a circular form, put it in the cytoplasm, and now you have extra chromosomal circular DNA. And this study here that was conducted in published in 2019 shows standing electron microscopic images of both chromosomal forms of DNA with different ranges of circular forms in DNA and different types of staining. So we do see biological forms of circular DNA that are commonly used to be studied. However, one thing that I'm really fascinated by is the circular forms of DNA that can occur because of protein binding. In particular, the kind of protein that I'm interested in goes back to the level of DNA organization that I had mentioned. Those beads on a string are histone core proteins that can coil the DNA in about 1.5 to 2 turns of DNA that wraps around it, and it's non-sequence specific. If I was to have some form of DNA and I have these histone core particles, there's going to be some degree of interaction that will occur. The studies that were conducted under Ariel Purnell in the 80s and 90s showed that different structures can be made based on the number of histone core particles that can wrap the DNA. As you can see here on the bottom, that shows more zoomed in approaches to the circular forms of DNA. You would see in options B and C, there's one histone core particle that wraps into 2 different types of DNA, a more open configuration in B and a more closed configuration. And open and closed has to deal with how the entry and exit free DNA either crosses itself or doesn't cross itself. And you see in D and E here that you can even coil 2 histone core particles on top of each other. So there is a biological model that we can work with and study and go deeper in not only just the structure, but is there any type of sequence-based influence? So for the seminar, I want to talk a little bit about what I've been doing in studying many chromosomal circles at lengths less than 500 base pairs. And that length is very important because genetic expression in chromosomes, they exist in many million base pair steps. I'm interested in very small circles of 500 base pairs and below, especially with the fact that statistical physics has showed us that there is a limit to the ability or the ease at which these circular forms can be made at like sizes of 150 base pairs or below. I want to talk a little bit about some of the software that we use, especially in DNA modeling and elastic energy minimization calculations. Give some insights that we've had in recent studies of our nucleosomal DNA. And a nucleosome is DNA that wraps around a histone. So that terminology, when we talk about a nucleosome, it's simply the histone DNA combined unit. And I want to talk about 2 studies that I conducted myself on looking at the effects of DNA length in the mini chromosome, as well as breathing of the nucleosome. And we talk about breathing as a way for adhered DNA that's on the histone to maybe peel back and no longer be bound to it. The way in which we do this is difficult in the fact that when we talk about computational simulations, we have to marry detail and information with computational expense. There's a lot of different directions that we can go into in studying in computational simulations, studying nucleic acids and proteins, anywhere from Monte Carlo calculations to molecular dynamics. And molecular dynamic simulations are amazing. It can account for all of the atomic coordinates of DNA, as well as interactions between the two. The downside is that it's so computationally expensive to run that you're looking at, trying to do an MD simulation only on the terms of like a nanosecond. And even that, if you use a high-powered supercomputing cluster, could take days to run. So we have this balance to figure out in terms of simulation work. How can we get quality data without loss of generality? What kind of approximations can we make so that when we do these studies, we can get some quality information? We know that one base pair has approximately 40 atoms, and that's a lot of detail to factor in. But if you take a look at this linear form of DNA, you might notice that every base pair between the two chains, they kind of look the same. They kind of look like a rectangular form. So we can model forms of rectangles on these base pairs. And if you notice also, those backbone chains also kind of move in the same similar direction. Now, this is a B form of DNA. B form DNA is one of many forms that exist biologically, but B form is the most commonly observed form. So we can make some approximations with that and say, well, if it's B form DNA and the backbone chains are relatively similar, I don't need to model that in our calculation. What I have now is a series of what I consider to be Lego blocks oriented in three-dimensional space. But what's great about this is that we are able to go from all of these atomic coordinates and atomic interactions to simply say, what if we had a set of variables or a set of parameters that only looked at one base pair orientation relative to its neighbor? And when we do that, we actually get 3 rotational parameters known as tilt, roll, and twist, and 3 translational shift, slide, and rise. And this is 3-dimensional. So I could have the same value be used to represent twist, and I would still create a 3-dimensional configuration. And I don't know about you, but if I had to choose between a level of organization, either with this file that just shows approximately 3 bases here on the left, versus working with structures that, 12 base pair steps are here, on the right-hand side, I have a lot less information in the right-hand side in terms of having to account for interactions, but I'm still able to generate a quality 3-dimensional structure that not only gives us properties in terms of base pair orientation and base pair step orientation, we can still work with sequential information as well. So by utilizing this, this rigid body parameter, setup, we can create 3-dimensional models, such as, let's say, a linear form of DNA, where we only care about uniform twist and uniform rise, which is a vertical displacement, and twist is just like rotating about the z-axis. If I had all the twists and rises the same value, I would get the structure that you see here in blue. And if I wanted to, say, make a 1 parametric change, like if I wanted to decrease our slide parameter, well, I would still get a 3-dimensional configuration. If I wanted to increase roll, which is the way in which one base pair opens or closes to the grooves of the DNA, I would get a different type of configuration. So we get a lot of wonderful information by just having representative parameter values. And if you're ever interested in taking a look at a more detailed information about how you can construct models of DNA, and maybe go into looking at A forms or C forms or the really weird Z form DNA, you can visit our website at web.x3dna.org, which is a wonderful web browser interface that allows you to create structures, learn tutorials, and see how they're connected with what we can study in terms of viral DNA, which is very popular with COVID-19 studies, as well as RNA structures, which is an entirely different, crazy direction to go into. So you're welcome to check out. And that's something that we in our lab have developed and continue to work on as well. My mentor, Dr. Olson, is actually a leader in developing this parameter orientation of parameter usage for 3-dimensional structures. And we can use this mathematically in looking at elastic energy minimization calculations. If you all know about calculus, we know that if we want to do some sort of optimization, we would need some sort of initial function or initial structure, as well as some sort of rest state to look at. Well, we can collect energy information at every base pair, as long as we know what the base pair rigid body parameters are, what the rest state parameters are, and some sort of elastic constants matrix that we can look at. And we can increase or decrease the level of sophistication of our force field. And when we say force field, it's the combination of our intrinsic rest state parameters, which we can model as a straight line if we wanted, and the elastic constants matrix. Now, this is a collection of interactions between translational and or rotational parameters in terms of stiffness or flexibility. If we wanted to model a very stiff polymer, then we would just sit here and say, well, maybe our bending parameters are very high, or our twist parameters are very high or low if we wanted something that's flexible. So we can model observations based on the dimer, based on the sequence, and we can model all those observations in terms of stiffness as well as orientations. Now, when we look at studying computational models of DNA and doing our elastic energy minimization calculations, we're really more interested in what's occurring in the rotational parameters, such as bending and twisting. And the reason that is because a lot of experimental work has been done on DNA in solution, particularly the persistence length, that stiffness that I had mentioned earlier. We typically associate that with bending persistence and twisting persistence. So what we do in terms of this larger function on the bottom is that we only account for changes in terms of rotation. We don't really care about relationships between rotation and translation, but we do have levels of knowledge-based force fields that give us that information as we go deeper into studying effects of sequence. And we want to set our translational parameters to be so high that they don't influence the overall optimization. So what I'm really getting at is, yes, we can go through and have high level detail to our optimization, but we can make approximations and go into just focusing in on representations relative to a linear piece of DNA based only on rotational and parameter changes, which is really nice for us because all we really want to try to do is understand what the structure is doing, that mesoscale approach that I had mentioned earlier. So as I mentioned with calculus, as long as we have a rest state and as long as we have our initial condition, we can go forth and do whatever type of minimization or optimization we want to conduct. So let's just say that I have this structure, this DNA bean that I made as my initial set. I have my force field. I can load it into our optimization program that we have called EMDNA. And we can have a level of control in our software that allows for us to maybe, just optimize it so our initial state looks like the rest state. Or we can hold some things in place to say, all right, well, let's just keep the ends frozen in space and allow the path of DNA to optimize and see what that kind of structure looks like. Or we can go one step further and say, well, rather than just the positions in space, let's also freeze the configuration of the steps at the beginning and at the end. So we have high level of control in how we want our optimization to run. But not only that, we can model protein DNA interactions in this as well. Because as I mentioned, we have that fixed positional and rotation of the fixed base pair step orientations. Well, what if that little curve that I'm showing in this structure is actually modeling protein DNA behavior? We can get all of that information from repositories like the Protein Data Bank or the Nucleic Acids Data Bank and say, well, this protein, when bound to DNA, looks like this. We can create that model and say, during optimization, let's freeze that and optimize everything else. And we can look at the influence that that blue region has on the free DNA that's coming before and after it. So this is how we look at that influence of a protein interacting with DNA and how that bound DNA will influence the way free DNA is going to configure. And this is all elastic. I could, if I wanted, incorporate some sort of electrostatic control because DNA is a naturally occurring negatively charged macromolecule. So I could include electrostatics in the calculation. I could have any different types of environmental studies if I had a highly negative solution or a highly positive solution. And what you see here is an example of an ideal form of circular DNA. We don't mess around with the conditions. All we want is just give us a nice, smooth optimization. We get a circular structure here. Now, when we talk about circular DNA in terms of our simulation, it's very important that we have the proper definition for it. So what we're saying here is that for circular DNA, our positions of the first base pair and last base pair must be identical. So that way we have that virtual base pair step that makes the circle become a circle. If we didn't have that, that overlap of the first and last base pair, then philosophically, we really wouldn't have a circle. So it's a way in which we have that level of, work in our simulation to get what we want in terms of circular forms. And I'm really excited to announce that today, the work that was done with my mentors, Dr. Olson and Dr. Cleveland, had their paper accepted in the Journal of Biophysical Chemistry today. So if you're interested in more information about our code and how we can do emDNA, we recently had that publication happen, which is nice. It's always nice to get a publication, and I'm going to be the one that is the point person for this, which is very exciting for me. What's really wonderful about looking at structures, as you can see here in this graphical abstract, is that there are different parameters that we can look at simply because we have circular forms. And those are topological parameters known as the linking number, which is an integer value descriptor that tells us how the two curves of DNA, the two helical curves, or the two backbone chains, are interwound in its form. And we have a parameter known as a relaxed linking number that takes into account what the number of base pairs we have in our sequences relative to how many base pairs are in one turn of DNA. And it's combined, is partitioned between two parameters known as the twist and the writhe. So the way that the centerline curve twists in relation to one of the outlying backbone curves, and then the writhe, which is just how the helical axis is deformed. So if you take a look at this structure, this EM structure that we got from Kronberg and Baker, you would see a circular form of DNA that has different levels of writhe associated with it. But if the linking number was the same for all of these, as it should be, because the linking number isn't able to change on its own, linking number is invariant. And the only way that linking numbers can change for a structure has to be that a protein has to come in and physically change by the orientation by either nicking one chain or both chains of DNA. So you can relax or constrain it, but typically if you allow the form of DNA to exist naturally without any type of external influence, the linking number will always stay the same. So maybe the linking numbers here are all the same, but we have different levels of writhe. And because of the way that this is partitioned, it's all based on how the base pairs are twisting about itself. And what's important here is that there's different levels of supercoiling that exist, such as negatively supercoiled structures or positively supercoiled structures. And most of the structures that exist naturally are negatively supercoiled. So we want to take a look at optimized configurations with respect to some degree of supercoiling, because those are more naturally occurring. So with all of these parameters in place, we can start to build up our study, and we can look a little bit closer at the nucleosome and how it pertains to the mini chromosome. What this animation shows you is that nucleosome configuration that I mentioned. We have a wrapping of DNA anywhere from 140 to 145 bases, 147 bases of DNA that are wrapping around a histone core octamer. And the different colors tell you what types of histone core particles are associated with it. So we have a dimer of H3 and a dimer of H4, and it looks almost as if it's two cylinders that create this wedge on top of each other. But if you look closer, you would notice that there's not it's not uniformly bound. It's like bound in pockets. So you have an interaction between the protein and DNA, and then you would have a region of DNA that doesn't adhere, and then another protein interaction and so forth. So it's almost as if there's a consistent pattern of binding and non-binding in this nucleosome. My friend and colleague, Dr. Chidoli, who's in my lab, he went through and actually did some wonderful work and some data science techniques to try to look at the collection of nucleosomes that do exist in the protein data bank now and look at particular conditions such as the level of resolution of our structures, and was able to get about 140 of these deposited nucleosomes. And if you notice in this collection here, they all follow in some way, shape, and form a superhelical path that's pretty similar. Then he wanted to go a little bit deeper in what we're looking at, because since we do see that the ends of these nucleosomes vary in space, there might be some sort of rhyme or reason why. So in the analysis, he first looked at the changes of our superhelical twist over 140 base pairs. And in the calculation, some structures had high levels of supercoiled twist and low levels. And if we go deeper, he went deeper and looked at how the individual bases have these degrees of superhelical change, and notice that they kind of partition itself or organize itself into different clusters. And if you can go a little bit deeper at certain positions, such as negative and positive 20 and 50, there are clear demarcations that occur in these nucleosome configurations that basically give us these twist cluster groups. twist cluster groups of of overwound or underwound configurations, which is really easy for us to work with because we're looking for insights into how nucleosomes, which are bound DNA, bound forms of DNA, could influence neighboring DNA. And we have ways to go a little bit deeper and to see, well, what does something that has a lot of overwound pieces of DNA structurally, how does that compare to something that has underwound configurations? And we can do this through different levels of mini chromosomal optimization, but we first got to construct the mini chromosome. And previously what was done was in the older coarse grain models, what they did was they just modeled a cylinder to wrap the supercoiled DNA that's supposed to replicate nucleosomes. They have a specific pitch. You model it around a cylinder, and then you just adhere a bunch of beads to the free loop. And they were able to construct this. And there are new ways for us to construct it, such as a Monte Carlo calculation. And we want to use this because it allows us to use real forms of the nucleosome that we can get from the protein data bank to create these mini circles. Yeah, you don't really need to worry about the mathematics here. What's really important for us, though, is that when we use this Gaussian Monte Carlo, the issue is that you can't really make a circle. You are going to have a level of gap between the first base pair and the last base pair. We can try to get as close as we want to creating this circle. As you can see in this structure here, there is a gap. So the initial state that we have is very important because maybe having a larger gap between the first and last base pair is the difference in optimized states. So we can use Gaussian Monte Carlo constructs, but we can also look at, there's some, I think somebody just had something in the chat room. We can also look at ways in which we can construct the mini chromosomal circles using a Bezier curve. And I'm really excited to share this with you because we had a student from University of North Carolina, Chapel Hill, named Abigail Watson, an undergraduate mathematics student who worked with us this past summer and specifically designed these mini chromosomal loops with this Bezier curve, which states that if we know the entry and exit points of our nucleosome, and we know the orientation of those DNAs, we can create any type of loop that we want. It could be narrow, as you can see from the construct on the right, or it could be flatter. But she was able to come into our lab and create this algorithm that allows us to customize a fully closed circular loop. So we have a plethora of options that are at our disposal in terms of creating these structures. I have a question here that says the general winding has to do with active production of certain gene proteins. And actually that's correct, Bryn. Different levels of winding could be the result of different types of proteins. Certain proteins are gonna really open up or cause that DNA to not have a high degree of winding anymore. So you might have a localized change in that DNA, and that's gonna influence the free DNA around it. So the loops could be looser, it could be tighter. It all depends on what's occurring in that local region of bound DNA. It could make the supercoiling worse. It can make the supercoiling more relaxed. So that comment, Bryn, that's in the chat room is absolutely right. So thank you very much for that statement and that question. So going along with this, continue on in the presentation, I wanted to talk a little bit about what I was able to do with my studies. So I have this schematic of what a mini chromosomal circle looks like. We have our free to move DNA in yellow, which is at the top, and we have our more bound nucleosomal DNA in red. And one thing that I can investigate is what would happen if we change the overall size of the mini chromosome, but we keep the nucleosome the same. As Bryn had mentioned, there could be different ways of unlocking or increasing or decreasing the supercoiling, and that might have to be a function of the overall length of our mini circle. But there's also something that we can look at where, what if we kept the overall mini chromosomal circle the same length? But in fact, we can change how many base pairs are actually binding or simulated binding to our DNA, to our protein, and it becomes free to move optimized DNA. So we have two different directions that we can go in this study. So the first thing that I did was I wanted to take a look at reducing the length of the mini circle. So I have different ranges of lengths of my circle, anywhere from 292 base pairs to 336, but I wanted to keep the length of the nucleosome the same, and I really only wanted to look at what was occurring elastically. I didn't want to take a look at what was occurring electrostatically because I just wanted to see what occurs specifically in terms of changing in length. And the way that I did this was I used the mini circles that I made from the Gaussian Monte Carlo calculations, and I needed to figure out a way for us to do these removal of base pairs as as symmetric as possible. So it was an alternative removal of base pairs from these blue regions. So I would remove a base pair from, let's say, the entry blue region, optimize, then remove from the exit blue region, and I would keep doing that until I got to a lower size of our base pairs. And what I noticed was there was an interesting pattern in terms of an opening and closing that occurs, whether I've removed an integer number of turns or half integer turns of our DNA. So for every integer turn of DNA that was removed, I noticed that I have a closed configuration in this particular under-twisted nucleosome in purple that also tends to replicate with this over-twisted nucleosome in gold. So I had this repetitive closing and opening of our loop. But one thing you might notice is that if I rotated the structures, as you can see on the bottom, I again have that closing and opening, but I have something that's inconsistent. If you take a look here at the T1 half and T1 for our under-twisted nucleosome, we have more of a needle shape, more of an open configuration on both views. But for the over-twisted nucleosome, I have a much more open structure in this profile view, but I have a closed view of this crossing of DNA in this side profile view. Why is it that a more over-twisted nucleosome produces this configuration while a more under-twisted one produces a more narrow open configuration? And if we go a little bit deeper, we can take a look at what the elastic energy profiles look like. And what's interesting here is that as I show this, as I increase the number of base pairs in my mini chromosomal circle, I have this periodic behavior in my energy, where I have regions of high energy versus low energy. And we want to shoot for low energy. Low energy biologically means a more stable configuration. What is interesting in this figure to us was that the more over-twisted nucleosome that was bound to our model had higher ranges of, had a higher variation in energy as you increase the size of our loop. While the under-twisted really, it did show a variation, but not as drastic of the two. Now, what we see here in the gaps here, there are two different sizes here. When you look at elastic configurations and you don't account for electrostatics, you run the option of an overlap in the structure. So biologically invalid structures. So we have two structures here that, since we just applied an elastic, we don't actually allow them to, or we don't actually account for them because they overlap on the centerline curves. So we can't allow them in there. But if we wanted to go a little bit deeper in the analysis, we can partition out bending contributions and twist contributions in the loop. So what's really accounting for the elastic energy? What particular parameter is causing it? When we go deeper, what we see is that with the under twisted on the top, significant energy is the result of the bending of that free loop. And then as you see this periodic behavior where we see where the closed structures are, closed and open variations in the energy doesn't seem to deviate much in our from the models that we showed earlier along the path here. And most of it's bending energy. The maximum and minimum are purely bend. So when we see the high energy state or the low energy state, twist doesn't seem to factor into that optimized energy state. However, when we look at the structures that we have from the over-twisted nucleosome that we used, we have significantly more often occurring contributions because of our dimer twist. So we keep the same nucleosome structure size of 141, but because of the parameters of the nucleosome core particle or the core structure, having a more over-twisted nucleosome results in influence of the loop at different sizes. which we think is, I think, is absolutely fascinating to see that the size does play a role, but really the level of orientation of the nucleosome structure also influences how the distribution of energy comes to be. It's mostly bend, which makes sense in terms of the persistence links that we find from the polymer, but twist plays a larger role in one over the other. So we wanted to take a look after this. We wanted to take a look at what would happen if we start to relax the number of base pairs that are considered to be adhering to the histone core particle. We kept the number of base pairs the same at 359 base pairs. It might be larger than the model that I had shown earlier, but 359 base pairs is the size of base pairs that we saw from the Ariel-Purnell study that I mentioned earlier. We want to stick with what biologically known lengths of DNA, and we want to make changes to the topology, levels of degrees of supercoiling, as well as the number of base pairs that we would consider to be the nucleosome, anywhere from 111 base pairs to 141. This time, we want to have a level of electrostatic control so that whatever our supercoiling diversity is maintaining that same level of supercoiling at the end. So anything that has a linking number, say, of 31, maintains that linking number of 31 after our optimization. So we want to try to keep that supercoiling as consistent as possible. This is important to us based on previous work that was done in the late 90s about Monte Carlo calculations that try to replicate breathing with the number of wraps of the nucleosome. As I mentioned, if you recall that cylinder I had mentioned, if they had reduced the number of base pairs that are modeled after that cylinder, what they notice is that there's different behaviors in the level of writhing in the overall structure. So based on different linking numbers, we would have different patterns of writhe based on how it's wrapped. And as I mentioned, we're interested in the length of 359. So if you take a look at some of the observations we got from the Swagone paper, we see that for a highly wrapped structure of 136 base pairs, we'd either have a highly closed structure for a linking number of 33, or closed but more open in the loop in this orientation for a linking number of 32. If they unwrapped it, say to 116 base pairs, they're both open in configurations. So we do see that from previous studies, there is an influence in wrapping. So we wanted to take a look at all of those nucleosomes that we had, apply that to the Bezier curve calculation function that we showed, run the optimization for well over 5,000 structures, and start to look for comparisons between the two. And one thing that we needed to find was an additional parameter to help us understand the influence that the nucleosome has on the loop. So here you see a mini chromosome model of DNA. In yellow is our nucleosome, and in gray is just a free loop of DNA. One parameter that we can find is a gamma angle, which is the relative orientation of the center of our loop relative to the anchor points of the free base pairs, which is where we have that junction on the nucleosome. So we can create a plane between the entry and exit base pair and a midpoint of the loop. And there's just a different orientation angle that you can see that we made this plane between these 3 points. We can find the angle between that plane versus the directionality of that midpoint. So we have a vector that we get from the directionality of that midpoint base pair reference frame, and then the orientation that it has with this loop. So we have this, and this helps us determine if we have a more open or closed configuration. And we can use this as an additional parameter to see, do we have more open or more closed relative to, say, energy. So I created a plot that showed all of the elastic energy values that we could get relative to the number of base pairs versus this open configuration parameter. And one thing that we noticed here was this clustering that was occurring based on what the linking numbers were. The y-axis is our elastic energy. So higher energy structures that you see at the top here in peak go towards a linking number of 37, which is a +3 supercoil. And as I mentioned, nature typically likes negatively supercoiled structures. So when we do this simulation, this optimization calculation on maintaining a highly positively supercoiled structure, of course, our energy states are going to be much higher. If we look here at our lower energy in the cluster in red, the cluster in gold, and then the cluster in green, the cluster in green is our lowest energy state. But what's interesting here is that represents linking number 33. For the size of DNA that we're working with, a more relaxed or more open configuration of DNA, the linking number should be 34. So our analysis shows that optimized states tend to like a configuration of a linking number that's at about -1. And what I like about that is that that's also what Ariel Purnell found in their studies for their many chromosomal circles. The likelihood for circles to exist at the -1 configuration is significantly higher than other configurations. So we're finding optimized structures whose energies match what we've seen biologically. But if you notice in that green region here, it seems like there's clustering in two regions. There's this side gap here on the left and this major cluster on the right. So we could go one step further and say, well, we know about this twist cluster groups, cluster 1 and 2, based on higher or under or over twisted clusters. Well, does that play a role in how the energies are dispersed? And it turns out it is. We see in that cluster of linking number 33 that a particular cluster in group 5 is a higher energy state than cluster group 3 and cluster group 6 and cluster group 7. So this is really interesting to us because not only did we see energy behavior based on the structural supercoiling diversity, but also based on the type of nucleosome clustering that we had observed. So we wanted to go a little bit further and try to see, well, okay, we know what the 3 lower energy states are. They were associated with linking number 32, 33, and 34. What would be the likelihood that they should exist biologically? So here we have 3 examples that are modeled on top of each other from cluster group 3, which is in purple, which doesn't really have a lot of excess, a lot of underwound or overrun twists. Twist cluster group 5 is in green and 6 is in orange. So you would see that linking number 32 is closed with a really nice open loop. 33 and 34 kind of has this mixed state. Some of them are closed and some of them are open. But if we were to look at the probability, if we want to take a look at the partition function to see, well, what's the likelihood that these are going to occur the likelihood that they would occur based on energetics, what's interesting is for twist cluster group 5, linking number 32 and 34 is likely to occur, which is different than what I mentioned earlier when the Purnell papers and the studies mentioned that linking number 32 is the predominant, more favorable state. For a particular type of configurations of the nucleosome, particular mini-circles are likely to occur in different supercoiled states, which if we look back to previous studies that we saw with the Monte Carlo calculations, we did see this likelihood of states between 34, 33, and 32, but this is where that wrapping comes into play. Would we see this level of probability if we start to relax what we consider to be our nucleosome? Let's start to increase the number of base pairs in our loop, decrease the number of nucleosomes the number of base pairs in our nucleosome to see if we can get this level of likelihood to continue. So here is a plot that relates our relative openness, gamma on the bottom, with a Writhe parameter. And if we recall, Writhe is just the level of distortion of our overall configuration. The x-axis is the number of base pairs that we start to peel back to go from a nucleosome to free DNA. And you'll notice that the Writhe starts to increase to 0, so it starts to become more open as we decrease the number of base pairs that are in the nucleosome or the number of base pairs that we are peeling back. We also notice that the gamma angle starts to decrease a little bit, but you might notice that the figure in orange here, which represents the configuration, the nucleosome that's the more over twisted, it maintained its Writhe and gamma orientation. It seemed as if it didn't want to open up compared to its examples from a more under twisted group or a group that had no excess twist. So I could take a look at the ride and look at the models every, let's say, 4 base pairs removed. And if you pay attention to the orange configuration, the orange structure, you'll notice that it maintains a closed configuration for as long as possible up until about a total length of 129 for the nucleosome. So it maintains this closed configuration. And in fact, it looks like it's kind of shifting in space as much as possible. It doesn't want to go away from that closed configuration up until a specific number of base pairs, and then it opens up. But even then, if you peel back to having a nucleosome length of 121 base pairs, a more unwrapped nucleosome, the structure itself isn't as open as the other 2 examples, which is fascinating to me, because if we open up this more, it should fall into similar structures because you're decreasing the level of wrapping to it. So if we focused in a little bit more on this and looked at the probability of states, we see that while the under-twisted or the no excess twist examples maintain a linking number of 33 as expected, what we saw was as that over-twisted state starts to decrease in the number of base pairs that are adhered to the nucleosome, the likelihood that it could exist as more than one topological state increases to about 50-50. But it shouldn't, from what we've seen experimentally, which I think is so cool, is that what we're seeing with the mathematics, the physics is telling us technically these 2 configurations can exist biologically based on what your parameters are. So why is it that biologically we're seeing the prevalence of one over the other? And we can start to go a little bit deeper now into studies of our breathing to maybe see what the influence of sequence could be. Maybe there's a sequence of that loop that would actually increase the likelihood of one type of state over another. One fascinating thing I want to point out right before I released this seminar a couple months ago was we knew what the configuration states looked like for linking number 33 and 34 for a particular number of base pairs that are unwrapped. So I know the structures of, let's say I unwrapped 20 base pairs. I knew what those structures looked like. So one state's in pink, one state's in blue. What was really cool was the previous Monte Carlo studies that were done in the 90s showed the exact same configurations, but in different configuration states. So this more open configuration in magenta for 34 that we calculated should be what is for 33, this optimized state in 33. But the nice thing about this is that the way we calculated our optimization versus the way that was done in the past was they were looking at one configuration of the loop. They were looking at the entire loop itself. We're optimizing at a base pair level. So by optimizing individual base pairs along the loop, finding these ideal configurations, re-optimizing and continuing, we have a higher level of control and a higher level of sophistication to our optimization state. And yet we're getting structures that looked similar to what we've seen in the past. The question is, how can we go deeper and see, well, maybe there's something to our findings versus a simple, previously established calculation. How can we go deeper and really understand what's going on? So in conclusion, we use this mesoscale approach that allows us to both model and optimize different configurations of protein-bound and protein-unbound DNA without losing generality, but also making sure that it's computationally inexpensive. We're looking at many chromosomal optimization configurations, so we have an idea of how nucleosome configurations influence upstream and downstream forms of DNA. And we've seen that more over-twisted forms of that nucleosome does significantly play a role in how those free linker DNAs, or those free upstream and downstream DNAs, influence their configuration, influence their elastic and electrostatic properties. We've seen that loop length is going to influence the optimized energy state, but there is now a question of whether or not there are certain topological families that might be more prevalent than the other. We didn't really talk about that with the loop length study. And lastly, the over-twisted nucleosomes tend to maintain linker shape at the start of unwrapping, but there is a less likelihood that it's going to change as we start to unwrap. And the question now becomes, is that a consistent property for an over-twisted nucleosome? Or is it something that's specific to that one nucleosome itself? So to conclude, I want to just take a moment to thank a lot of the people that have got me here in terms of my research support team here at Rutgers. And here in the middle, you see my mentor, Dr. Wilma Olson. My friend Steffi's in the background, and my colleagues, Dr. Pamela Perez, who got her PhD a couple years ago. Now she works as a contractor with NOAA. My friend Syed is finishing up his PhD in physics and hopefully will be defending this year. I have collaborators at Baylor and the University of Leeds in the United Kingdom that have really helped shape me as a researcher. We also have opportunities for undergraduates. And I will say that the COVID response and the shutdown of a lot of research didn't happen for us. We were computational. The only thing that had to change was our rigs had to go from being at the office to being at home. So being able to do computational research during this time hasn't really hit a snag. And it's actually been able to offer undergraduate students, not only from Rutgers, but from places like Chapel Hill, the opportunity to do computational research. And I want to point this out to you that if you are interested in doing research, don't waste that opportunity, especially if you're just now trying to figure out what it is that you might be interested in. If you're interested in any of the things that I mentioned to you today, all you have to do is email me, ask some questions, and know that computational research is a direction that is at hand. When I was at App, I didn't have a lot of opportunities to explore computational research because I really thought physics was simply experimental. And being able to do computational studies opened up an entire new world for me of what our physical understanding is and how we can explore that in different regimes. Lastly, though, I want to thank everybody at Appalachian State. I might choke up, so I apologize if I start to get a little emotional here. I started at App, never had taken a class in physics. I actually am from Maiden, North Carolina, which is south of Hickory, so if you know where Maiden is, great. But I never had a physics class until the first day of undergrad. And Appalachian State changed my life. I actually think I got a better undergraduate education at App than the people that here are at Rutgers or at neighboring Princeton or TCNJ or Columbia, because y'all have faculty that truly wants to be there for you and develop you as amazing physicists. I tell people here all the time that I'm on first name basis, and I still talk to everybody at the faculty at Appalachian. People here at Rutgers barely talk to faculty members. They typically talk to postdocs or they talk to graduate students. You have an amazing cadre of faculty at Appalachian, and I would not be where I'm at without them. Like Tanya Coffey, Christopher Thaxton. I even learned a lot from Brooke Hester, and I say that because she just started as I was leaving, and I never took a class from Dr. Hester, but I had opportunities to learn what she was working with the magnetic tweezers to get started. But I could not be where I'm at without Dr. Jennifer Burris, who I am kind of hoping is here. Hi, Dr. Burris, she's in here. Dr. Burris is the reason why I'm here. So I thank her so much for the mentorship that she continues to give. And I want you all to know, as students at Appalachian, please take advantage of every minute that those professors want to be there for you, because they see something in you that you might not. Dr. Burris did. And to this day, I cannot call her Jennifer. I can only call her Dr. Burris. But those faculty members want to see you become the best physicists or chemists or biophysicists that you want to become. To every single faculty member listening, this research, my dissertation, my master's and everything, it's all your fault. You're to blame for all of this. And at this time, if you have any questions, I'm happy to entertain them. Thank you so much for listening. And again, thank you so much. So I have a question here from Bryn that says, how would your computational program work with current genetic computation, like with the next generation Sanger-based genomic reconstructions? That is a wonderful question. And I had mentioned earlier that I'm looking at sizes of about 500 base pairs. Our model is specifically centered around more configuration states, more of the physics behind things. Once you get to a certain size limit, let's say 500 to 700 to 1,000 base pairs, the program starts to break down. And when we start talking about current genetic computations like high C or high throughput kind of sequencing, which is like thousands and thousands of megabases, our program doesn't really work. So in terms of what we look at with genetic computation, our program doesn't work in that regard. But there's a catch. The catch is, what if we were looking at genetic computations in terms of, let's say, building a database of circular forms of smaller DNA for cancer biology? We have a collaborator that we work with in our lab a lot at Johns Hopkins University, and his name is TJ Ha. And he's created this database of small circular forms of cancer-based DNA. And we could actually add to that database with our program that says, okay, well, if we know the sequence of a particular size of DNA, we know that sequence, we know that size, and we can start building more sophisticated sequence-dependent models, well, we can contribute to that, at least to have a genetic understanding of circle to gene. And that's actually something that I'm very passionate about. A part of my dissertation that I haven't talked about is the role that sequence plays on structure. And I'm actually in the middle of compiling new force field information from the protein data bank, which is celebrating its 50th anniversary this year. So I'm excited about what new information that we can get in terms of sequence dependence. Bryn, that is an amazing question. Thank you so much for asking that. I had a question here or a comment here from Court that says, great talk, excited to see how the underlying sequence influences your outcomes. Court, me too. And the cool thing about sequence dependence is that we can start small and build our way up. We can see a lot of sophisticated, dimer specific or even tetramer specific, force fields. Or we can just say, what happens if we have a particular AA step? And how does an AA step differ from everything else? So we have a lot of control that we can play with there. And I'm excited to see what we can do with that. Dr. Chandra, I just want to let you know that I can see your lips move, but I do not hear you, but you are not on mute.
Here, I'll take over for a minute. It looks like Matt Phillips has his hand raised. Matt, do you want to ask a question?
I was just curious, how computationally expensive are those Monte Carlo simulations that you run?
So I'm able to do about a 150 base pair circle in about 12 seconds. I had just done it because I had to recompile my entire code today on the build that I have for my interface. And if I go back, let me just go back one quick second. Here, the slide, I mentioned that one base pair is about 40 atoms. So imagine imagining how to account for 40 atoms over 40 interaction vectors for one base pair, let alone 150. So if I wanted to run I ran a very simple calculation where it was, okay, here's a circle, 150 base pairs. I just want to compare it to ideal linear DNA. Took me like 12 seconds at most. So it's not expensive at all. The actual programming itself, we use a lot of C++ code. It takes up maybe 25 megabytes. So I don't have to worry about a lot of space here. And the one thing that I think is really great is that when we use supercomputing clusters, I can run 13,000 of these simulations all at the same time, and I'm done in an hour without hurting cluster space. So it's not very expensive at all compared to what you would see with molecular dynamic simulations.
It looks like Sean had his hand up too. Sean, do you want to ask your question?
Yeah, thank you very much for sharing your research with us. This might be more of a chemistry question or like biology question, but like take a topical example like COVID-19, the protein on the outside of the virus cell. Once you find out what that protein is made of, is it purely like geometric how it affects DNA, and is it like how do you work like a new protein that is discovered into a model like this? Is it
That,
mostly geometry, or is there like a lot of chemistry involved?
that's a great question, and it's actually it falls on a lot of the work that's being done by some brilliant experimentalists who do work on X-ray diffraction, cryo-electron microscopy, which got a Nobel Prize a few years back. When we look at the interaction between a protein and DNA, it can be geometric, but usually it's going to be electrostatic. It's going to be based on the electrodynamics that exist between the protein shape and the DNA. It typically is a mix of the two. Predominantly, we're looking at electrodynamics mainly, but geometry does play a role, especially the geometry of DNA. And that's something that I didn't touch on. I mentioned that degrees of supercoiling are more prevalent in biology, but I also said that DNA and proteins, they don't have a brain. So if a piece of a protein comes in a contact or is in a region of DNA, it's trying to find a gene, but let's say that gene is knocked out based on geometry, the electrodynamics or the electrostatic signal is out there, electrochemical signal is out there, it just can't physically get to it. So it's this relationship between geometry and electrochemical gradients that are, it's like a 1-2 punch. You can't have one without the other. And there's a lot of work that's done with molecular biology where they try to knock out as many of those variables as possible, which is why circular forms of DNA are so common in molecular biology labs, because you knock out geometric steric issues. So they try to keep everything as open as possible. So it's easier for the protein and the DNA to interact with each other, especially when you look at, I literally have lost my train of thought here. When you look at NMR studies or nuclear magnetic resonance studies or cryo-EM studies, you really want to try to minimize those variables as possible. But yeah, that's a great question. And it's typically a combination of the two. One is more influence than the other just based on how heavy the protein might be or how complex the protein might be.
Dr. Chandra, is your mic back? Doesn't look like it. Okay, so so I just want to be mindful of the time. I know it's 5:04, so I'm going to say that folks can go if they want to go. I think there's one more question left in the chat that I'm sure Rob was happy to answer.
So I got 2 questions in chat right now. One question from Charlie says, would these sort of simulations have any luck with questions about protein folding, misfolding as well? And that's a great question. These particular types of simulations could be. The question is, how do you want to model it? If you have, let's say, 17 models of proteins that have interactions with DNA, and they all have varying levels of folding or misfolding, but they still have a level of DNA interaction, I can take a look at it and say, all right, well, if I have those structures and I have those models of DNA, I can upload that to some sort of structure that I make, and then I can see the influence on the optimized of DNA. So it does have that option to look at it, but that's kind of not the overall approach to it. But you can, if that's the direction you wanted to go to. Let's say I want to take a look at what I mentioned back to what Sean had mentioned about COVID-19 and those and the spike proteins. Well, if let's say that they're doing a protein folding experiment with cryo-EM with 30,000 variations of protein folding, but they all have a level of DNA interaction, we could look at that as well.
All right, anybody else have any questions? Okay, well, this is what I'm going to say real quick because Rob is amazing, but I'm going to tell y'all why he's amazing. He's amazing because he is one of the hardest workers of any students that I've ever known. And so I'm telling you all of that because we talk about how amazing people are a lot, and people seem to assume that they're amazing because they have some sort of natural ability of amazingness. And I think it's important to recognize why Rob is amazing right now, and it's because he's a damn hard worker. And so I just wanted to mention that because there's a lot of people chatting in the window that know Rob from long ago about how amazing he is, but that's why he's amazing, because he's one of the hardest workers I've ever met. So
one of the things that Dr. Burris spent years trying to get out of my head was I never felt like I belonged. I never felt like I was smart enough. But darn it, if I wasn't going to be the hardest worker out there. Sometimes, being diligent and spending, like, every day in the library until 2 a.m. I might not be naturally gifted at something, but I'm not going to let anybody work harder than me. That's really where a lot of the work came from. And also having Dr. Burris breathing down your neck for, assignments and research, deadlines isn't going to, hurt the process.
I'm not that bad, am I?
No, you're not. Of course, you're a mate. No.
Okay, everybody. I hope you have a wonderful weekend. Thanks for coming.
Thanks, everybody. I hope you have a